In this video, we are dealing with the topic of SDS-PAGE. SDS-PAGE is a molecular biology method used for the detection, separation, and analysis of proteins. What does SDS-PAGE stand for? SDS-PAGE stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis, which is a polyacrylamide-based gel electrophoresis technique that uses SDS in the sample preparation. During an SDS-PAGE, protein molecules are separated by their size within an electric field. SDS-PAGE can be used for qualitative or semi-quantitative protein analysis. For proteins to migrate through the gel based only on their size, denaturation and masking with SDS are required. Proteins in their native or natural state are folded in various globular forms with different sizes and surface charges. To prepare the proteins for SDS-PAGE, they are incubated in a mixture of SDS, beta-mercaptoethanol, or D-thiotetriol at 95 degrees Celsius. Heat denatures the protein, causing it to unfold. Beta-mercaptoethanol, or DTT, help break internal disulfide bridges, while the now completely denatured and linearized protein binds to the negatively charged SDS ions. SDS coats the entire protein, masking all charges on the amino acid chain. The number of negatively charged SDS ions that bind is proportional to the protein's size, with 1.4 grams of SDS binding per gram of protein. Consequently, larger proteins carry a greater negative charge than smaller ones. This causes smaller proteins to migrate faster through the electric field towards the anode. In sample preparation, glycerol is added to increase the sample density, making it easier to load it into the wells. A dye, usually bromphenol blue, is also added to track the sample's migration throughout the gel. SDS-PAGE is considered discontinuous because the gel consists of different layers. These layers include the stacking gel and the resolving gel, which differ in their degree of polymerization and pH. The stacking gel is coarse with a lower acrylamide content, while the resolving gel is finer with a higher acrylamide content. In commercially available SDS gels, the resolving gel may even have a gradient in the acrylamide concentration. The stacking and resolving gels serve different purposes. As the name implies, proteins are stacked or concentrated in the stacking gel. At the boundary between the stacking and the resolving gels, proteins of different sizes converge before migrating uniformly into the resolving gel. Let's first look at the stacking gel with a pH of 6.8. In this system, glycine and chloride ions are present. Chloride ions are negatively charged at pH 6.8 and smaller than the proteins, allowing them to move quickly toward the anode in the electric field. Acting as leading ions, they migrate ahead of the proteins through the stacking gel. Glycine, on the other hand, is partially positively charged at pH 6.8, resulting in a low negative net charge. This causes glycine ions to migrate slowly through the electric field, trailing behind the negatively masked proteins. Glycine serves as trailing ions, moving more slowly than the proteins. Between the leading and trailing ions, a voltage gradient is established, leading to an increased electric field strength in the space between them. This causes the proteins to migrate quickly and concentrate at the boundary of the resolving gel. Once the proteins and ions have been concentrated at the interface between the stacking and the resolving gel, they can enter the resolving gel. The prior focusing sharpens the bands because all proteins and ions enter simultaneously. The resolving gel differs not only in acrylamide concentration but also in pH. At pH 8.8, glycine ions carry a negative net charge, increasing their speed in the electric field, allowing them to overtake the proteins. The glycine ions now follow the chloride ions, which continue to migrate the fastest. Without the influence of trailing ions, the proteins can separate evenly by size within the electric field. Since the resolving gel is more finely meshed, larger proteins move slower throughout the gel, while smaller proteins navigate more easily through the matrix. If the goal is to study the properties of native proteins, native PAGE is preferred over SDS PAGE. Native PAGE preserves the physiological properties of proteins, making it possible to study phosphorylation or separate monomers from multimers. A separation occurs based on the isoelectric point and folding of the proteins. Unlike SDS PAGE, proteins are not denatured or masked, so they retain their native shape and charge. This means that proteins of the same size but different charges migrate at different speeds in the electric field, due to differences in net charge. Proteins of the same size but different folding are also separated if their folding differs, for example if the protein is globular or denatured. In native PAGE, large negatively charged proteins migrate the slowest through the gel.